Cloning refers to a number of different processes that create biologically identical entities. Examples of cloning in nature include some plants and single-celled organisms that reproduce through asexual reproduction. This happens when you usually have one parent and it goes through mitosis to create a clone with identical DNA. Examples of cloning in humans are twins, which are created when you take a fertilized egg and you split it to create two humans that are biologically identical. And this, according to learngenetics.utah.edu.
According to genome.gov, there are 3 different types of artificial cloning. There is gene cloning, therapeutic cloning, and reproductive cloning. In gene cloning, certain DNA segments or genes can be isolated using genetic engineering and used to fight hereditary diseases such as hemophilia, cystic fibrosis, and diabetes. In therapeutic cloning, researchers produce embryonic stem cells that can be used to test drugs, learn about diseases, and grow healthy tissues. This is done by researchers cloning embryos and waiting about 5 days for the cells to go through mitosis to the point where there are about 100 of them. This is called the blastocyst cell stage. And each of these cells can become anything in the body, and they can be, as I said before, grown to into healthy tissues to replace diseased ones. This can be used in the future to grow pieces of liver, heart, and other important organs. Reproductive cloning is important because this is the cloning that would be used to create humans. The last type of artificial cloning I would like to talk about is reproductive cloning. This is often done through somatic cell nuclear transfer. Somatic just means a body cell, or a cell that has the organism's normal number of chromosomes. Now, brown bears have 74 chromosomes. And in somatic cell nuclear transfer, you need 2 different organisms. You need a surrogate mother, who will actually supply the egg and grow the clone, and you'll need a specimen. This is the animal that you're cloning. So from the surrogate mother, you'd get an egg. This would have 36 chromosomes, and you would get a somatic cell from the specimen. This would have 74 chromosomes. Now you would take out the nucleus of both cells. You would throw away the nucleus of the egg cell. Throw it away. Then you take the nucleus of the somatic cell, you implant it into the surrogate mother's empty egg cell. So now you have a full egg cell with the right number of chromosomes. Now, for this cell, researchers would then stimulate it electrically to make it think it's been fertilized. Then, after about 5 days, mitosis would happen, and there would be enough of an embryo to implant back in the mother, and she would actually give birth to this clone.
The history of cloning begins in 1885 with Hans Streich. He had 2 identical sea urchin cells, and when he shook them, he created 2 genetically identical sea urchins. This is the first example of artificial embryo twinning. In 1952, modern cloning begins with Robert Briggs. He used nuclear transfer and took the embryonic cell of a tadpole and put it in the surrogate mother of a frog to create clone tadpoles, which never quite reached adulthood, but this paved the way for future experiments. In 1984, Steen Willesden created the first mammal through cloning, which was groundbreaking, by using embryonic nuclear transfer. In 1996, Ian Wilmot created Dolly, the first mammal created by somatic cell nuclear transfer, where you take a body, adult, somatic cell, and you put it in instead of having to use an embryonic cell. In 2007, Shukrat Mitelapov created the first primate embryonic stem cell, and he used somatic cell nuclear transfer, which ended up paving the way for therapeutic cloning that we can use in humans to repair kidneys, hearts, and other things like this. In 2013, Shukrat Mitelapov he did human embryonic stem cells that he created by somatic cell nuclear transfer. So this is just kind of how the point we are right now. And we got this information on learn.genetics.utah.edu.
Our stance on cloning is that cloning is bad. Cloning is bad because it's experimental. And in the many experiments done with cloning, it has been shown that that cloning presents higher risk of disease and death in organisms, that is highly inefficient, and that it brings about a host of social issues. To back up the claim about susceptibility to disease and death, the FDA did a 2006 risk assessment of animal cloning, and they concluded that cloned animals suffer from a host of deformations and diseases, such as liver, heart, and kidney problems, as well as head deformities and immune system problems, and many other issues that kill almost a third of all neonates. And the clones that survive past that time live a significantly reduced lifespan. To back up our point about the inefficiency of cloning, we look at the high death rates previously mentioned, and also genome.gov states that cloning is a highly inefficient process in which very few clonees grow up to become healthy individuals. Genome.gov cites Dolly, and with Dolly, the sheep in 1996, it took 277 attempts in which only 4 to 5 sheep actually made it into the surrogate mother, and of that, only Dolly survived. And also, in the 2007 study published by the National Biotechnology Information Center, a 150 cow study, 42% of them died. These high death rates and high rates of disease make cloning very, very inefficient. To back up our point about social problems, in 2001, the National Bioethics Advisory Commission released a statement saying that SCNT undermines important social values and open the door for eugenics that allows people to tamper with who is created and what they look like. And this goes back to history with people like Hitler, who wanted a master race, and it allows people to manipulate people like objects instead of people. This is really important to the development of us as a world.
One of the biggest arguments for cloning is bringing back a loved one. One of the biggest examples of this is bringing back a child. According to worldindata.org, the mortality rate has been dropping for decades, but the mortality rate was still stuck at 4.3% by 2015, which means millions of parents could potentially clone because they missed their child. However, when looked at this with logic, this argument falls apart. According to the National Biotechnology Institute Center, the environment that children are raised in impacts their health and their knowledge and personality later in life. This is much like as in a twin. Two twins will look the same, but their personality and their experiences will be extremely different. So your clone might look the same as your previous child, but they will have very little in common elsewhere. Dr. Ian Wilmot, senior scientist in cloning Dolly, said, "Each child should be wanted as an individual, and if you set out to make a copy of someone who has already been here, you are saying that you want the new baby to act like that person, which is not fair to do to a child."
In the words of the great Bill Nye,
Cloning is a tricky business. Now, if we have this much trouble with farm animals, are we really ready to try it with humans?